Hi everyone, good morning. Hello. Hi everyone, good morning, good morning, good morning. How are you all doing? Am I perfectly audible and visible? Tell me, am I perfectly audible and visible? Good morning, Trisha. So tell me everyone. Okay, so welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to continue with the same chapter. I know we have stretched this chapter a bit, but that's fine. It's okay because we have to cover this class 12th syllabus in detail. It is important for your boards as well, right? And when it comes to the class 11th, you know that we are going for the detailed marathons, isn't it? Yes, we are even going for the detailed marathons. Fine. So there is nothing to worry about before NEET, before your board exams. We will finish your syllabus, and yes, you will get sufficient time to revise as well. But what you have to do is you need to be consistent. It is the consistency which is the key. That's all. Remember it, right? So now you can see, yes, we are going to continue with the same chapter, but moreover, see, this is what you need to focus. Tomorrow at 6 p.m. I will be taking breathing and exchange of gases, and it is a detailed marathon. Okay. Tomorrow at 6 p.m. in the evening, right? So I'm sure you'll be free in the evening. So we are going to cover this chapter, that is breathing and exchange of gases, and it is going to be a one shot. So yes, you can expect that class will go for 4 hours, 5 hours, or maybe for the 6 hours, right? So be prepared for this chapter as well. And you know that already we have covered digestion and absorption in 2 parts. This one we will cover in 1 part, and along with that we will also practice the questions. Okay, so see you all here in this particular session, and kids, join Avengers 2.0 as soon as possible. Please do not delay, because there also we are covering the human physiology. After finishing human physiology, we will go for the syllabus of class 12, and in class 12 we are going to start with the genetics. Fine. So most important units we are covering right now. So 70% of the classes will be live, and 30% will be in the recorded form. And in the month of November, we are going to provide you the holidays so that you can cover that backlogs as well. So there is a complete plan for you. All you need to do is you need to join this batch. So join it. Link will be there in the description box of any video on our channel, and just join it and be the part of this wonderful batch as soon as possible. Okay, as soon as possible. And yes, I know many students are requesting for the Plant Kingdom part 2. I don't know what happened to that particular video, right? So don't worry. See, let me finish this human physiology then, because it is for the revision, right? It is for the revision. So let me finish the human physiology. I'll keep one separate session for the complete Plant Kingdom. Okay, I can see again and again students are asking the same thing. What about Plant Kingdom? What about Plant Kingdom part 2, part 2? Array, whatever I have completed, at least cover that now, right? You'll get you will get session on that too. Fine. Okay, so here now you guys can see, just look at it. Fine. So here on this channel you'll get everything. See, your physics strategy, about all about the NEET examination, how to revise quickly, and see here also I have shared the detailed strategy with all of you. Okay, so do check it out. Fine, do check it out. Done. So now let's go, students. Let's start the chapter, and yes, let's start the session. So you know that in the last class we were talking about the female gametophyte, isn't it? Isn't it? We have discussed everything related to the female gametophyte. So now in the chat section, quickly tell me what is that female gametophyte. Yes, everyone. Quickly let me know in the chat section what is that female gametophyte. Quick. What is that female gametophyte? Quick. Yes. Quick. Embryo sac, are you sure? Are you sure? Yes, you are right. So let's start the story from that ovule part. Okay. So you know that the ovule, right, covered with the integuments, right? And here you know that the part which is not covered with the integument, it is the micropylar end, right? It is the micropilar end, and just opposite to the micropilar end, here you will see the chalazial end. Which end will be there? It is the chalazial end. And now here, this tissue in between is the nucellus, right students? This tissue present in between, what is it? This tissue is your nucellus. Just a minute, let me... Yes, so the tissue present in between is what? It is the nucellus. It is highly nutritive. It is going to provide the nourishment basically, right? What will it do? It is going to provide the nourishment to the developing megaspore. So you know that here from this nucellus, one cell, your archisporial cell, right? It will grow, it will divide, isn't it? Isn't it? Let's quickly revise it. It will grow, it will divide, and that archisporial cell, right, will form parietal cell, sporogenous cell. And that sporogenous cell will start behaving like the megaspore mother cell. Okay, right, it will start behaving like the megaspore mother cell, and then it will undergo meiosis. So you know that in the case of females, what is going to happen? From large megaspore mother cell, only one functional megaspore will form. What will form? Only one functional megaspore will form, right? So 3 will get degenerated, only 1 will be functional, and this functional one will grow and it is going to form the embryo sac. You know about it. Am I right? You know about it, right? So here in the nucellus, you will be getting this embryo sac. So near to the micropilar end, students, here you will be having egg and the synergids. Towards chalazial end, there will be the antipodal cells, and the largest cell of the embryo sac of the female gametophyte is this one. It is the central cell, right? Right? It is the central cell. Is that clear? Right? Is that clear? So here you can see this is the polygonum type of embryo sac. What type of embryo sac it is? It is polygonum type of embryo sac. This is what you need to remember. Okay? Okay, so today we need to start the fertilization part here. So before that, you should know about the cells and everything. Clear? Clear? So this is near to the micropilar end. Here you have the egg apparatus. So egg apparatus, it includes 3 cells, students. This one is the egg, and these two having filiform apparatus, these are the synergids. Clear? These are the synergids. And near to the chalazial end, here what do you have? You have the antipodal cells. What do you have? You have the antipodal cells, and this one is the central cell having 2 polar nuclei. Okay, having 2 polar nuclei. So any doubt? Yes, any doubt here? Sure? Do you have any doubt here? So next part is fertilization. This is what we need to revise today. Okay. Next part is the fertilization. So you know that after pollination, what is going to happen? After pollination, what is going to happen? You know that the pollen grain is going to fall on the stigma here. Am I right? The pollen grain is going to fall on the stigma here. Am I right? Yes, students. Am I right? Make the class interactive, everyone. Just discuss it in detail. Today we have to finish this chapter. Today we have to actually finish this chapter. So please focus here. So this is going to happen if this pollen grain, right, if it is having the compatibility here, you know that the pollen grain will germinate here, right? It is going to germinate here on stigma. Am I right? Right, it is going to germinate here on the stigma. And what is going to happen? This pollen grain, it is going to absorb the moisture. It is going to absorb the moisture and nutrients from this stigma. Do you know that? What is going to happen here? Here, this pollen grain, as I said, this pollen grain is going to, yes students, this pollen grain, it is going to absorb the, it is going to absorb the moisture from the stigma. Okay, it is going to absorb the moisture from the stigma, and along with that moisture, some sugars, some carbs will also be absorbed by the stigma. Then what is going to happen? Basically pollen grain will absorb moisture and stigma from the stigma. Moisture and sugar will be absorbed by this pollen grain. So what is going to happen here, bache? It will swell. This pollen grain will swell. So I hope you remember the structure of, right? I hope you remember the structure of your pollen grain. So you know that when we talk about the pollen grain, right, the plasma membrane is there, no doubt. Then comes the intine and then comes the exine. Do you remember this part? Then comes the exine. And when you talk about this exine, you know that you will see the germ pores. Am I right? What will you see here? This exine is discontinuous. It is having the germ pores. Do you remember that? It is having the germ pores, right? So what are the germ pores? The exine, the area of exine where sporopollen is not deposited, right? This part is known as the germ pore. Okay, so for your quick revision, remember dicots, they have 3 germ pores. Monocots, they have 1 germ pore. So dicots, because they have 3 germ pores, they are tricolpate. Monocots, because they have 1 germ pore, they are monocolpate. So I hope you remember this part. So no doubt when this pollen grain, it will swell from this germ pore, what is going to happen? The pollen tube will form. What is going to happen? The pollen tube will form. And in this pollen grain, right, you know that it is 2-celled stage. In 60% of the angiospermic families, it is 2-celled stage. Remember this? It is what? It is 2-celled stage. So it is having one large vegetative cell, also known as tube cell. Fine. And the smaller one is what? It is the, yes, smaller one is what? It is the generative cell. Remember? It is the generative cell. So finally this pollen grain, after absorbing moisture and sugar, it will swell up and the growth of, right, this pollen tube will start. Fine, bache, the growth of this pollen tube will start. And let me tell you one very important point here. So for the pollen germination, please write down. For the pollen germination, boron is required. Okay, actually even the calcium also plays the role here. Calcium and boron both are important for the germination of this pollen grain, but yes, the main role is of boron, right? The main role is of boron. So it is an important question. Fine, bache, it is an important question. Is that clear? Yes. So 60% of the angiospermic families, they have the pollen grains which are 2-celled stage. These are the 2 cells. So finally they will show the growth. And which element is going to help here? It is the boron, right? It is the boron. Is that clear? Yes, bache, is that clear? Sure, sure. And now when this pollen tube will germinate here, okay, this pollen tube is going to form some enzymes also. Do you know that? Do you know that? When this pollen tube, it grows, okay, when this pollen tube, it grows, it forms the enzymes, and that enzymes are going to help to digest the solid style here. You know now this receptive part is stigma, this tube-like structure is style, the swollen base is the ovary, right? So if the style is solid and the pollen tube, when it is growing, when it is germinating here, so it is going to release some enzymes, and that enzymes we are going to, you know, eat the surrounding tissue. Fine, bache, the enzymes are going to eat the surrounding tissue. So that's how it grows. Clear? Clear, bache? Is that clear? So, and it is not mandatory that always only one pollen tube will form. Sometimes in the member of some families, like I'll give you the example of Malvaceae, right? Here you will see polysiphonous condition. Right, what type of condition you are going to see here? You will see polysiphonous condition here, that the pollen grain, it will form many pollen tubes. So it is not mandatory that always it is going to form one pollen tube. No, it can form more than that as well. Clear, bache? It can form more than that as well. So this part clear? Yes, this part clear? Sure? Sure, 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 sure. Yes. So ultimately when this pollen tube is growing downward, so what is happening here? Let me show you. So now this pollen grain is germinating. So now when this pollen tube is coming downward here, first of all, you will see the vegetative nucleus. Which nucleus will you see? You will see the vegetative nucleus, also known as tube nucleus. Clear, bache? Also known as tube nucleus. You know that the role of vegetative cell here is to form the pollen tube. What is the role of vegetative cell here? To form the pollen tube. And now, bache, the generative cell after it. Right, the generative cell, the nucleus of generative cell, it will divide here mitotically, and it will form 2 male gametes. Are you getting it? It will form 2 male gametes. Are you getting it? So what is going to happen? The pollen tube, the generative cell, sorry, the vegetative cell is going to form the pollen tube. So here you will see the tube nucleus, and then what will be there? This generative cell will divide mitotically. It will form 2 male gametes. So when the pollen grain is in the 2-cell stage, then on stigma, right, that generative cell will divide to form male gametes. And when that pollen grain is in 3-celled stage, already, right, already it is having 2 male gametes and 1 tube nucleus. Any doubt? Right? Right? So it will be having 2 male gametes and 1 vegetative nucleus. Is that clear? Yes, everyone. Is that clear? Sure? Sure, sure, sure. Okay. Any doubt here? Yes? Done? So ultimately, you know that this pollen tube is going to, basically what is the main motto here? It will grow, it is going to, right, this pollen tube, ultimately what is the main motto here? The main motto here is that this pollen tube, right students, this pollen tube is moving towards the ovule. Right, ultimately the egg is present. Pollen grain is a male gametophyte. Embryo sac is a female gametophyte. And you know that that egg is present in that female gametophyte, in that embryo sac. So obviously this male gametes, because they are also non-motile, isn't it? So where are they going to reach? They will reach to that embryo sac. Clear, bache? To that embryo sac. So this is what you need to remember. And actually this movement is again, chemotactic. Right. So now you know the structure here. See, you know the structure here. You know that here, when we talk about the egg apparatus, we have this egg and we have the synergids. Yes or no? Like this is the egg here. And here you have the synergids. Now, bache, the synergid is having the filiform apparatus. What is it? It is having the filiform apparatus. These synergids. What do they have? They have, just a minute. Yes, they have these are the synergids, and here they have the filiform apparatus. So, bache, actually that pollen tube, it grows towards these cells, right? Right? These cells, these synergids, these filiform apparatus, they will release some calcium boron inositol complex, right? What will they release? They will release some calcium boron inositol complex, like some chemicals will be produced by it. What is going to happen, bache? Some chemicals will be produced by it, right? So that is why the pollen tube is growing towards these synergids. Clear? The pollen tube is growing towards these synergids. Are you getting it? This is important. Okay, this is important. The pollen tube is growing towards these synergids. Why? Because here they are releasing some chemicals. That chemicals are actually that calcium boron inositol complex, right? Calcium boron inositol complex. Clear, bache? Clear, bache? So now this part, the germination of this pollen grain here and the entry of this pollen tube in the ovule, right? All this process, the germination of the pollen grain on stigma and then the entry of that pollen tube in the ovule, this process up to this part, it is actually the pollen-pistil interaction. What is it? It is actually the pollen-pistil interaction. Is that clear, bache? What is it? It is the pollen-pistil interaction. Done? Pollen-pistil interaction. Clear, bache? Yes or no? Germination of pollen grain and then the entry of pollen tube in ovule. This complete process is what? It is the pollen-pistil interaction. So that's what you need to keep in your mind. And the ideal temperature for the germination of your pollen grain is 20 to 30 degree Celsius. Clear? Optimum temperature is 20 to 30 degree Celsius for the germination of the pollen grain. Clear? Clear, bache? Any doubt? Yes? Any doubt here? Tell me, any doubt here? See, this is the diagram. So see, many pollen grains, they are trying to germinate, but the compatible one will grow and the pollen tube is going to enter here, right? It is going to enter here in the ovule. So this is basically the pollen-pistil interaction. Okay, this is actually the pollen-pistil interaction. So you can see here, this part, right? And yes, it is also important. So let me highlight this part. So actually, firstly, here you will see bache, male gametes, and then there will be the tube nucleus. Okay, firstly there will be the male gametes here, and then there will be the tube nucleus. It will be followed by that tube nucleus. Like these are the male gametes, right? Because we have to follow the NCERT, you know that. And this one will be your tube nucleus. Okay, this one will be your tube nucleus. Is that clear? Is that clear? Tell me, is that clear? So see this diagram. Just look at this diagram, right? So see, the pollen tube is entering here in the synergids. Here you have this filiform apparatus which is releasing the calcium, boron, inositol complexes. Actually, these are the sugar complexes, right? These are the sugar complexes. So here you guys can see that male gametes, and then here you are having the vegetative nucleus. So ultimately, bache, so just imagine it like this, like chemicals will be released by that filiform apparatus, by that synergids. Pollen tube is growing towards it. It is coming like this, right? It is coming like this. Clear, bache? So what is going to happen? Ultimately, when that pollen tube will enter in that embryo sac, right? When it will enter in that embryo sac, what is going to happen at that time, right? The synergids, they will break, right? They will get degenerated. You can see it here. They will get degenerated. Can you see it here? They will get degenerated, and then all the content of this pollen tube will be released here, like male gametes and the tube nucleus. Can you see this part? Yes, can you see this part? One by one we will discuss it from NCERT as well, but first of all understand this diagram, right? See, it is written here also, discharge of male gametes into the synergids and the movement of sperms, one into the egg, another into the central cell. Right, it is showing that. So they can simply give you the diagram, and then they can ask you that what this diagram is representing, right? What is happening here in this diagram? So whenever you read the NCERT, consider yourself as an examiner, okay? That you are going to set the NEET paper. Fine, that you are going to set the NEET paper. So the diagrams here are also important. Clear, bache, Trisha? Sure? Done? So, see this part. So please open up your NCERT and please highlight the important terms here, okay? Kindly highlight the important terms here. So, pollen-pistil interaction. Pollination doesn't guarantee the transfer of right type of pollen, right? What is the meaning of right type of pollen? The compatibility should be there in the stigma and the pollen grain. You know that, right? Right? So genes are also going to decide that, and moreover sometimes because that air current is bringing the pollen grain, so it is not mandatory that each and every stigma will receive the pollen grain of same species. Are you getting it? It is not mandatory that stigma is going to receive the pollen grain of the right stigma is going to receive the pollen grain of the same species. It is not mandatory at all. Okay, bache? Okay, bache. So here you guys can see that pollination doesn't guarantee the transfer of right type of pollen, that is the compatible pollen of the same species on stigma, right? So often pollen of wrong type, either from other species or from the same plant, they also land on stigma. Okay, so pollination is not ensuring that fertilization will occur. Clear? Pollination is not ensuring that fertilization is going to occur. Right, bache? Right, bache? So the pistil has the ability to recognize the pollen, whether it is of right type or of the wrong type. So ultimately it is the pistil that will recognize us. Is that clear? Question can come from this part as well, right, bache? And then it is the right type of the pistil that accepts the pollen and promotes the post-pollination events, okay? Post-pollination events that will result into the fertilization, okay? That will result into the fertilization. Clear, bache? Clear, bache? Right? So if the pollen is of wrong type, pistil is going to reject it. It will not germinate, bache. Right? So ability of pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and the pistil. So ultimately it is that interaction, right? The pollen grain and the pistil. Of course, genes are going to play the role here. Right? When it comes to this compatibility, the genes are going to play the role here. This is what you need to remember. Okay, this is what you need to remember. Clear, bache? So chemical components of the pollen, they are going to interact with those of the pistil. Of course, even the chemicals, even you know that it is basically defined by the genes. Clear, bache? So it is only in the recent years that botanists have been able to identify that some pollen and pistil components and their interaction, it is leading to the recognition followed by the acceptance or rejection. So this is what you need to keep in your mind. Done. So some students were saying, na ma'am, we want NCERT reading. So bache, in every lecture I always keep the NCERT with me, right? And I always used to explain the things from the NCERT. Done. And in this way you have to read your NCERT. Understand our concept, read it, right? Read it twice and then highlight the important points. Okay, then highlight the important points and revise that highlighted points again and again. And sometimes, you know, we do not have the sufficient time to revise the complete chapter. So in that case, what you can do? Do you know what you can do? You can simply, right? You can simply read the summary. It will give you the quick recall. Okay, you can even read the summary of the chapters. Fine, bache. You can even read the summary of the chapters. Clear? Clear? So pollen, after that compatibility test, the pollen germinates on stigma, it will form the pollen tube, right, bache? So the content of the pollen grain, it moves into the pollen tube, right, bache? So pollen tube will grow through the tissue of the stigma and style and it reaches the ovary. Right, bache? So you know that some pollen grains, they are shed at 2-cell stage. I have already discussed it with you. So in such plants, the generative cell divides. It will form 2 male gametes during the growth of pollen tube. Okay, so it is the generative cell mitotically which is going to divide and it is going to form the male gametes which are non-motile. And when the pollen grain is shed in 3-celled condition, you know that in that case already male gametes are present from the beginning. Done, bache? Already male gametes are present from the beginning. Is that clear? Is that clear? Now here, bache, 3 points are there. See, they are saying that pollen tube, after reaching the ovary, it will enter the ovule through micropyle and then enters one of the synergids. Right? So it is always not mandatory that pollen tube will enter through the micropyle. Okay? So if pollen tube, right, if it is entering through micropyle, if pollen tube is entering through micropyle, then you will call it as porogamy, right? And mostly it is the porogamy. Clear, bache? Mostly it is the porogamy. Like pollen tube is entering through the micropyle, but bache, sometimes it is also entering through the chalazial end, right? Through that antipodal cells. Sometimes it is also coming like that. So then you will call it as chalazogamy, right? Then you will call it as chalazogamy. And sometimes this pollen tube is entering through integuments. Okay, it is entering through integuments, right, from the sides. So obviously in that case you will call it as mesogamy. Okay, in that scenario you will call it as mesogamy. So no doubt mostly it is the porogamy, but it is not mandatory that always it will be the porogamy. Fine? Fine. So this is what you need to remember. So it is written here now that pollen tube after reaching the ovary, it enters the ovule through micropyle. So if it is entering through micropyle, then it means porogamy is occurring here. Clear, bache? Porogamy is occurring here. Done, bache? Done? So many recent studies, they have shown that filiform apparatus present at the micropylar part of synergid, it guides the entry of the pollen tube. So it is the filiform apparatus that will guide the entry of the pollen tube towards the embryo sac, and you know that why? Because of the chemicals. Clear, bache? Because of the chemicals. So here you can see all that events from pollen deposition on stigma until pollen tube enters the ovule. They are referred as pollen-pistil interaction. Clear? They are referred as pollen-pistil interaction. Again, the question can come from this particular part. Okay, question can come from this particular part. Done, bache? Done, bache? So pollen-pistil interaction, it's a dynamic process. Fine, it's a dynamic process where your pollen recognition is followed by its promotion or inhibition. Right, bache? So that's what you need to remember. So any doubt? Any doubt here? Tell me, any doubt here? It's very easy. So here they have given the example that you can easily study pollen germination if you dust some pollen from flowers such as pea, chickpea, right, Protellaria, balsam and vinca on a glass slide. right? So you have to put the sugar solution about 10%. After that, for 15 to 30 minutes, observe the slide, right? And you guys can see the pollen tube coming out. So have you tried it ever in the lab, in the school lab? Yes. Do you know about it? Yes, Vartika, do you know about it? Sure? Sure. So after it, what we need to understand, see, let's go as per the NCERT. Let's talk about the artificial hybridization, and then we will be discussing the double fertilization. Now you know that we have limited topics, so we can finish it today, right? We can finish this chapter today. And at 12, I have a class in our Avengers batch. So let's be very quick. Fine. Okay, Soumyadeep, just give it a try. Once again, make sure that your sugar solution is that 10%. Okay, so next to it is the artificial hybridization. See, what exactly is the artificial hybridization? The same thing we will discuss even in the genetics, right? You know that the self-pollination is possible, the cross-pollination is possible. Sometimes, let's say you are interested and you want a particular type, right? Sometimes, let's say you are interested in, getting a particular type of flower or getting a particular type of plant, or you can say that you want to promote a certain trait, right? In the plants, let's say you want to promote the certain traits, that this is the trait that I want to be there in my plant. Clear, bache? So in this case, artificial hybridization is preferred. Okay, so it comes under the crop improvement program, of course. Right, it comes under the crop improvement program, of course. And here what is happening, actually, the crossing experiments are going to take place. What is going to happen here? The crossing experiments are going to take place here, where desired pollen grain, where on the stigma, right, we can spray the desired pollen grain, and basically we want that plant, that stigma, to grow that desired pollen grain. This is what artificial hybridization is, right? Artificially you want to cross two different plants, okay, two different plants, right? This is what you want. And it is not mandatory that crossing different species, like one student is saying crossing different species, even it is not mandatory, right? In the chapter strategies for enhancement in food production, you will talk about it, right? You will talk about it. But here, what is the artificial hybridization? That artificially you want a certain pollen grain to grow on the stigma of that particular plant. Fine, bache. So what is going to happen here? First of all, let's say, basically here we need to protect our stigma from contamination. We don't want that stigma to allow the growth of any pollen grain, any random pollen grain. We want a specific pollen grain to grow on that particular stigma. Clear, bache? Clear, bache? So first of all, here what we need to focus, what we need to do, see, the two techniques are there actually, right? One is emasculation, another is bagging. One is emasculation, another is bagging. So first of all, let's say if in artificial hybridization, if your flower is bisexual, right? If your flower is bisexual, then in that scenario, what is going to happen? Yes, in that scenario, what is going to happen if your flower is bisexual, right? You need to remove that stigma, you need to remove that anther, right? Because we don't want our stigma to get contaminated. So in that case, you have to remove that anther, and the removal of anther is the emasculation, right? So emasculation is only required when your flower is bisexual. Okay, emasculation is only required when your flower is bisexual. Otherwise, there is no need, right? So after that emasculation, of course, you have to cover that flower so that contamination, right? So that contamination cannot occur. So if the female parent bears bisexual flower, removal of anther will be there, bache, right? From the flower bud before the anther dies, right? So we can use it with the help of that forceps, and this is known as emasculation. Another MCQ. So removal of anther from a bisexual flower is emasculation. Then, bache, emasculated flowers, we need to cover it with a bag of suitable size, right? So you can use the bag made up of butter paper so that contamination can be avoided. Done, bache. So this is known as bagging. So after bagging, sometimes we tag also, right? So when the stigma of the bagged flower attains the receptivity, like when that stigma will become mature, right? So mature pollen grains collected from the anthers of the male parent, they are dusted on that stigma. Flowers will be rebagged and the fruit allowed to develop, right? So this is the artificial hybridization. Clear, bache? This is the artificial hybridization. Fine, fine. So next to it is the double fertilization, again, which is very easy. Let me tell you, when we talk about the double fertilization, tell me, is it there in the case of angiosperms only or even in the case of gymnosperms? Do we find double fertilization in the case of gymnosperms? Yes or no? Double fertilization is there in gymnosperms, yes or no? Tell me quickly. Tell me quickly. Do we have double fertilization in the case of gymnosperms? Yes or no? No. But double fertilization is there in the case of angiosperms only. It is not there in the case of your gymnosperms. Okay. In the case of gymnosperms, that is why, right? Some people are not aware of it. So please focus. In the case of gymnosperms, because there is no double fertilization, so you are not going to see no triploid endosperm will be there. Okay. No triploid endosperm will be there. This is I'm clarifying initially. There will be no triploid endosperm. And here actually the female gametophyte, further it will grow. It is behaving like it will be nutritive. It is known as the endosperm. It is haploid then. Clear, bache? So gymnosperms, they do not show the double fertilization. So what exactly is that double fertilization? Now see, I told you that in the pollen tube you will be having, right? You will be having two male gametes. Fine. You will be having two male gametes and there will be one tube nucleus, right? Which is responsible for the growth of this particular pollen tube. Now, when you look at that particular diagram from NCERT, right? When you see that diagram from NCERT, so you know that these male gametes, this pollen tube is growing towards that filiform apparatus. So ultimately these male gametes are going to enter there, right? In that embryo sac. So basically that synergid will get disrupted there. Okay, it will get degenerated there. Clear? Yes or no? Yes or no? So out of these two male gametes here, actually in double fertilization, two steps are there. One is the actual fertilization, or you can say that true fertilization, right? One is what? One is the true fertilization and another is the pseudo fertilization. And this pseudo fertilization is actually known as triple fusion. Fine. It is actually known as triple fusion. So now you know the structure of this embryo sac here, isn't it? Isn't it? You know the structure here. See, the pollen tube is coming. So one male gamete here is going to fuse this egg, right? It is going to fuse this actual egg and one male gamete will fuse this part. Okay, so what is this true fertilization? In the true fertilization, male gamete and the female gamete will fuse. Egg and ovum will fuse here, right? What will be there? Egg and the sperm will fuse. The male gamete and the female gamete, right? Male gamete and the female gamete fuses. This is what true fertilization is, right? This is what true fertilization is. And of course, when this one male gamete, when it fuses with the female gamete, it will result in the formation of the diploid zygote. Yes or no? It will result in the formation of the diploid zygote. Yes or no? So actually, this egg after this fertilization, it will be converted into the zygote. Right, this egg after the fertilization, it will be converted into the, this egg will be converted into the zygote. Clear, bache? And these synergids in the surrounding, they will degenerate. They will get degenerated. Okay. So now, at this another male gamete, it will fuse with these two polar nuclei. Right? It is going to fuse with this two polar nuclei. So that is why it is the pseudo fertilization. So one male gamete One male gamete fuses with polar nuclei. Okay, it will fuses with polar nuclei. So actually students, just remember one thing here. See, no doubt you know that central cell is the largest cell of the embryo sac. Are you aware of it? That it is the central cell which is the largest cell of the embryo sac. Tell me quickly. Are you aware of it that it is the central cell which is the largest cell, right? Which is the largest cell of the embryo sac. Are you sure? Are you sure? So now sometimes here what happened? No doubt now I'm showing you that N plus N condition is there. That there will be two polar nuclei. That's what I said now. That in central cell you will see two haploid polar nuclei. Two haploid polar nuclei. So bache, these polar nuclei, even they get fused and they form diploid secondary nuclei. Right? So even if you will read this word secondary nucleus, it's not nuclei, it's nucleus. Right? Do not get confused here. When these two polar nuclei, they get fused, obviously towards maturity, when they get fused, they will form diploid secondary nucleus. Clear? They will form diploid secondary nucleus. So even with that diploid secondary nucleus, when your haploid male gamete will fuse, right? With that diploid secondary nucleus, when your haploid male gamete will fuse, what is going to happen? It will result in the formation of a triploid nucleus, yes or no? It will result in the formation of a triploid nucleus, yes or no? Yes or no? This process is very easy. So ultimately after fertilization, right? And this is what triple fusion is, right? This is what triple fusion is. It is, you can even say the fusion of N, N and N, or simply you can say that haploid male gamete will get fused with the secondary nucleus, diploid secondary nucleus of central cell. So you can call it in any way. Done bache? Done bache? So that is what double fertilization is. Okay, that is what double fertilization is. And after this double fertilization, as I said, the egg, it will form zygote, the synergids will degenerate. Your central cell is now called primary endosperm cell and its nucleus is primary endosperm nucleus, that is PEN, right? And even the antipodal cells, they will get degenerated. Fine bache? Fine bache? So after fertilization of knee, what is going to happen here? That egg will form zygote, central cell will form primary endosperm cell. Is that clear? Central cell will form primary endosperm cell. Is that clear? Yes. Central cell will form primary endosperm cell. Clear bache? Clear bache? Tell me quickly. So diploid nucleus, which is the secondary nucleus, which is the definitive nucleus, it will get fused with one of the male gamete, that is your triple fusion, and egg will form with the one of the male gamete. This is your true fertilization, that is your syngamy. Clear bache? That is your syngamy. Clear bache? Clear, clear, clear. Sure? Done? Are you sure? Okay, so now here you can see one thing, that ultimately these non-motile male gametes, they'll come to the embryo sac with the help of pollen tube, yes or no? It is the pollen tube which is bringing non-motile male gamete towards the female gamete, right? So this process is known as siphonogamy. What is it? It is the siphonogamy, right? This siphon-like structure is formed. This tube-like structure will form. So it will take that male gametes towards the female gamete. Clear, bache? It is the siphonogamy. So if there is any doubt here in this part, do let me know. Okay, do let me know. So let's quickly revise it. So after entering one of the synergid, pollen tube releases the two male gametes into the cytoplasm. And how is it possible? See, when pollen tube, when it will reach that embryo sac, okay, actually the pollen tube will burst and even the tube cell will also degenerate at that point of time. Why? Because endosmosis is occurring. This pollen tube, endosmosis means excess of water will enter in that pollen tube. Because of that, it will burst. And then obviously, right, because when it will enter that embryo sac, what is going to happen? endosmosis, and that's why it will burst, right? So tube nucleus is degenerated. One of the male gametes will fuse with the female gamete. That is true fertilization, also known as syngamy. Fine. That is syngamy and also known as true fertilization. And it will result in formation of zygote. And other male gamete move towards two polar nuclei, or sometimes these two polar nuclei, they fuses, they form diploid secondary nucleus, right? They form diploid secondary nucleus, also known as definitive nucleus. Clear, bache? And you will get your primary endosperm cell. Fine. Even the NCERT is very easy. It is very, very, very easy. In NCERT, it is written in very easy language. Clear, bache? So, because there is the fusion of 3 haploid nuclei, that's why it is the triple fusion. Fine. That's why it is the triple fusion. Clear, bache? So, now we need to discuss the post-fertilization development events, or basically after zygote formation, what is going to happen. This is what we are going to discuss. Clear, bache? So what will we discuss now? Post-fertilization events. Fine. Post-fertilization events. Clear, bache? Yes? Tell me. Clear? Sure? So ultimately now we need to talk about the endosperm. Right? Now we need to talk about the endosperm. So see, zygote, and this one is your primary endosperm cell. Am I right? This one is your primary endosperm cell having triploid nucleus. Am I right? This cell, this large central cell, because now it has formed the primary endosperm cell and it is having the primary endosperm nucleus. So actually, bache, when you talk about the post-fertilization changes, first of all, we need to talk about this endosperm. Then we will see that how zygote will grow. Then we will see that seed formation and the fruit. That's how we are going to cover this part. Okay, that's how we are going to cover this particular part. Okay, bache. So now here what is happening actually? What is the thing which is going to take place here? Before the zygote development, it is the endosperm that will develop. Okay, that's what you need to remember. Before the zygote development, it is the endosperm that will develop. Why? Because this endosperm is nutritive. Okay, why? Because this endosperm is nutritive. The cells here are having the stored food, and that stored food is important for the zygote development. Clear, bache? That stored food is important for the zygote development. So before the development of zygote, the endosperm will develop so that it can provide nourishment to this zygote. Clear, bache? Clear, bache? So when you talk about the endosperm, you will discuss about 3 types. Okay, one is your free nuclear endosperm. Free nuclear endosperm. Another is your cellular endosperm. These are the 3 things that you need to discuss here. And another is your halobial endosperm. Another is what? It is your halobial endosperm. So what we need to discuss here? We need to discuss the free nuclear endosperm, cellular endosperm, and the halobial endosperm. Right? What 3 types are there? Free nuclear endosperm, cellular endosperm, halobial endosperm. And what is the ploidy of this endosperm here? Yes, Soumyadeep. What is the ploidy of the endosperm here? Tell me. What is the ploidy of the endosperm here? Tell me, tell me, tell me. Tell me quickly. Quickly tell me. Yes. Yes, bache, tell me quickly. Three type of endosperms, and I'm asking about their ploidy. You cannot even answer their ploidy. It's very strange. It's quite strange. Only one student is answering. Of course, it is triploid. Of course, it is triploid. When you are talking about the endosperm, its ploidy is triploid. Okay, it is going to be triploid. And if it is the gymnosperms, as I said, the female gametophyte will start behaving like the endosperm. So there it will be haploid in gymnosperms. But in angiosperms, it will be triploid. So we have 3 types of endosperm formation, right? What do we have? We have 3 types of endosperm formation. So as I said, it's very simple. It's C. It's free nuclei endosperm or free nuclear or simply nuclear endosperm, right? It's very easy. This portion is actually very easy. And then comes bache cellular endosperm. Then comes cellular endosperm, and then it is the halobial endosperm. Fine. It is the halobial endosperm. Now, when it is the free nuclear endosperm, actually what is happening, free nuclear or nuclear? Simply, see, if it is your primary endosperm cell, right, its nucleus will undergo karyokinesis, right? Only the nuclear division will be there. It is not immediately followed by cytokinesis. You know, this is how the process occurs. Like firstly there will be the karyokinesis, that is your nuclear division, and after that karyokinesis there will be cytokinesis. But here what is going to happen, because it is free nuclear endosperm, so initially only the nuclei will divide, right? And it depends, right, the number of nuclei, it varies here. So initially only nuclear division is there, only nuclear division is there. So thousands of nuclei will be present there. Clear bache? Thousands of nuclei will be present there. And how are they going to arrange? Let me show you here. Like see, if this is the structure, see in the center a kind of vacuole will form. These nuclei will arrange themselves towards the periphery, right? And when it comes to the maturity, towards maturity, what will happen? The cell wall formation will be there. Then there will be the cytokinesis. Okay, then there will be the cell wall formation. Are you getting it? Are you getting it? So initially it is just free nuclear, right? It is just free nuclear, right? Bache, only your nuclei, they are dividing. Later on at the time of maturity, some nuclei, they will arrange towards the periphery, and then the cell wall formation will start. Clear bache? And then it will get converted to the cellular endosperm, right? So the name is explaining everything. Free nuclear endosperm. Fine, free nuclear endosperm. And if I have to be more specific, then let me show you. So firstly endosperm development, and meanwhile here your zygote is also growing. Clear? Meanwhile here your zygote is also growing. So this is your free nuclear endosperm. And the coconut water, right? Coconut water of tender coconut. It is actually bache this free nuclei endosperm. Fine. Coconut water is nothing, it is the endosperm having so many nuclei. Clear bache? Having so many nuclei. And that white part that you talk about, that we eat in that coconut, that is the cellular endosperm. But when you talk about the coconut water, that is the free nuclear endosperm. Clear bache? That is the free nuclear endosperm. So coconut water, free nuclear endosperm, right bache? And when you talk about that white part, right, that is actually the cellular endosperm. Is that clear? So when it is cellular endosperm, of course, again here also the story is same, right? Here from the beginning you will see that endosperm is having the cell walls, right? From the beginning it is properly cellular. Clear bache? Clear? So here you are not going to see the free nuclear stage. Fine, here you are not going to see the free nuclear stage. Clear bache? So this type of endosperm is very common in the case of gymnosperm. So majorly it is there in the case of angiosperm, sorry, right? This is what you are going to find in the angiosperm. Now next is the halobial endosperm. It is a kind of intermediate stage, right? It's a kind of intermediate endosperm. So now bache, what is going to happen here in halobial endosperm? See, unequal division will be there. Your primary endosperm cell, it will divide unequally. Unequal cytokinesis will occur. What is going to happen here? Unequal cytokinesis will occur. And now later on, see, now this nuclei will divide. It will be like this. Now within these two cells, you will see many nuclei. Can you see that? Like firstly, the primary endosperm cell will divide. It will divide into two cells. Right? It will divide into two cells. Of course, they have acquired the cell wall, and then within these cells only nuclear division will occur. Clear bache? Within these cells only nuclear division will occur. Is that clear? Is that clear? So first division of primary endosperm nucleus is followed by unequal cytokinesis. Two unequal sized cells will be there. right? And then there will be the nuclear division, okay? So multinucleated condition will be there, and finally these nuclei, they'll form the cell wall around. Clear? They will form the cell wall around. Fine, they'll form the cell wall around. So this is about the type of the endosperm. So what is the function of endosperm here? It is highly nutritive. Okay, it is highly nutritive. It will provide the nourishment. And do you know that there are some angiosperms where your endosperm is not there, like your family Orchidaceae. Family Orchidaceae doesn't contain endosperm, right? It doesn't contain endosperm. So here you can see that endosperm development, it precedes the embryo development, like it is before embryo development, right bache? Right bache? So primary endosperm cells divide repeatedly. It forms a triploid endosperm tissue. So cells of this tissue are filled with reserve food material used for the nourishment of developing embryo. Clear bache? Clear bache? So this primary endosperm nucleus, it will undergo successive, like one after another nuclear division. It will form free nuclei. So this stage is the free nuclear endosperm. Later on cell wall formation will be there, then endosperm will become cellular. Clear bache? Clear bache? So coconut water from the tender coconut that you are familiar with, it is nothing, it is the free nuclear endosperm, and the surrounding white kernel is nothing, it is the cellular endosperm. So it is also the favorite question of examiner. Fine, it is also the favorite question of examiner. And this part, bache, I'll explain this diagram to you now. I hope you have seen, like when we started our channel, you remember we used to take the marathons for the NEET examination, and at that time, I was teaching you this, that exercise part. Your Wazeem sir was doing that, right? Yoga part. This is the yoga part that we are going to discuss. Fine. So that's what we have to cover now. So up to this part all clear? Yes, up to this part all clear? Sure? Sure. Okay, so this is about your endosperm. Okay, this is about your endosperm. So bache, after this now, let's cover the development of embryo. Then I will tell you about two types of embryo. Okay, so now what we have to discuss, we need to discuss the development of embryo, that how this embryo will further develop, like this part. This is what we are going to discuss now. Okay, this is what we need to discuss now. Clear bache? Clear? So what is it? It is the development of embryo. Right, now we know that the zygote will grow into embryo. So don't you think it is the embryogenesis now? Yes, don't you think it is the embryogenesis now? That from zygote, right, it is the development of embryo from zygote is known as embryogenesis, right? Is known as embryogenesis. Is that clear? Is that clear? What is it? It is known as embryogenesis. Clear bache? Clear bache? Right? Fine. So first of all, what will happen, bache? See, embryo will develop towards the micropylar end. I hope you know about it. I hope you know about it. Yes. Do you know this now? Yeah. Tell me. See, you know that the zygote is towards the micropylar end. Where is it? It is towards the micropylar end. So here your zygote will form, of course, right? And you know that it's a very important adaptation, that zygote development will start after the development of endosperm, so that it can provide the nourishment. Now just look at this diagram. Can you see the zygote? Can you look at the type of division, the first division that has occurred here? Can't you see, can we say that it is the transverse division? Can we say that it is the transverse division? See, if I talk about the zygote here, right? Wait. Yes, if I talk about the zygote here, of course it is diploid. So the first division that will occur, and here it is mitotic division, of course, bache. So this division is actually your first division is your transverse division, right? It is your transverse division. Clear? It is transverse division. So because it is here in the, like, imagine it, it is in the embryo sac, right? So this part is near to the micropylar end, yes or no? This part is near to the micropylar end, yes or no? So the cell which is near the micropylar end, you will call it as basal cell or the suspensor. Do you know that? This particular cell here, this one, it is the basal cell or the suspensor cell. And this cell here, it is the apical cell. And actually it is the cell which will form the embryo. So it is also known as embryonal cell. Clear, bache? It is also known as embryonal cell. Okay, it is also known as embryonal cell. So look at it. This is the zygote. It is dividing. Transverse division will be there. So here you can see two cells will form, basal cell and the apical cell. So the cell which is near to the micropylar end, right? The cell which is near to the micropylar end, students, this is the basal cell or the suspensor cell, and actually this cell, this apical cell, is going to form the embryo. So it is the embryonal cell. What type of cell it is, bache? It is the embryonal cell. Is that clear? Is that clear? Yes. So apical cell is also known as the embryonal cell. Clear, bache? Clear, bache? So both these cells, they are going to divide, right? Both these cells, they are going to divide simultaneously, right? Simultaneously they will divide. Like your basal cell will further undergo the division. It will undergo many transverse division. It will form a structure like this. And when you talk about this embryo, okay, this apical cell, so then it will form a structure like this. So ultimately, together it is the part of this, together it is the embryo, but here I'm just trying to say that this zygote will divide in such a way that here you will be having two cells, right? So both of them will divide simultaneously, but the type of division can vary. Okay, type of division can vary. Are you getting it? See this, type of division can vary. Can you see that? So here you need to remember the stages. See, first of all, when this basal cell is dividing, here you have this large cell. You used to call it as haustoria as well. Do you know that? We can also call it as haustoria. See, this complete portion is your suspensor. Am I right? This complete portion is your suspensor. Yes or no? Tell me, this complete part is suspensor. Please reward, bache. This complete part is what? It is the suspensor. Okay, it is the suspensor. Yes or no? Yes or no? Yes or no? Tell me, bache. It is the suspensor, this complete part. So actually the cell which is near to the micropylar end, it will swell. You will call it as haustoria, right? So here you will see the transverse division is there. 6 to 10 transverse divisions you will see in this basal cell, right? 6 to 10 transverse divisions you are going to see in this basal cell. But when you talk about this embryo, here you will see transverse plus the longitudinal divisions as well. Right, transverse as well as the longitudinal. So here you need to focus on the stages. Like initially you will be having pro-embryo stage, you will be having pro-embryo stage, then PG, globular stage, H, heart shape stage, M, mature embryo. Right, so here we have one trick to remember these stages. So question can come from this part. It is PGHM. What is it? It is PGHM, right? That initially it is the zygote. The zygote will form your pro-embryo, then globular, then heart-shaped, then mature. See, bache, you know that when you talk about the angiosperms, in the angiosperms we have the two classes, right? I hope you remember that we have the two classes. One is the dicotyledons, another is the monocotyledons. But the initial stages of this embryo formation, they are all same. Okay, Vartika, the initial stages are the same here. Initial stages are what? Initial stages are the same here, right? So this complete process is the embryogenesis. So I'm revising, repeating it. The zygote is dividing transversely. You will have two cell, basal and the apical cell. It is the apical cell actually. It is the terminal cell actually which will form embryo, okay? And this basal cell will just divide transversely. What is the main role of this basal cell? Actually, just imagine here this process is taking place. Okay, so the function of this basal cell is to, the function of this basal cell and this transverse division is to push the, right, to push the embryo towards the endosperm. What is the main role of this basal cell? What is the main role of these basal cells here? Is to push the embryo towards the, endosperm so that it can get the nourishment. Right, so this topmost basal cell, it will swell up. You are calling it as haustoria, and then this remaining part will be there, right? And then here you have the endosperm. So look at the stages here. So after zygote, you will say the pro-embryo stage, then globular heart-shaped embryo will be there, and then the mature embryo. So you should know the sequence. Okay, so initial stages are same for both the classes, for the monocots as well, for the dicots as well. So here you have the trick, that is PGHM, postgraduate hospital management. You can remember it in any way. Okay, for me it's postgraduate hospital management. Okay, so pro-embryo stage, globular stage, heart-shaped, and then comes the mature embryo. So just look at the structure. See this heart-shaped part. So we are calling it as cotyledon. This is the radicle here, right? So actually in between, see, so actually in between, let me write it here. In between radicle and plumule, what you have? You have the embryonal axis. The region between this radicle and plumule, we used to call it as embryonal axis, also known as tigellum. Okay, also known as tigellum. I'll explain it, right? I'll explain it so you can note down. Right, so this area or axis between plumule and radicle is embryonal axis. So it is the tigellum. Okay, it is the tigellum. Clear, bache? The main embryonal axis. Clear, bache? Clear. So here we need to talk about the, so as I said, the initial stages are same, but later on it varies. So you know that we have two classes. One is the dicots, another is the monocot. One is the dicot, another is the monocot. So bache, now here the point is di. Di means two cotyledons. What are these cotyledons? These cotyledons are nothing. They are the reserved food. Okay. These cotyledons are nothing, it is the reserved food, right? So if you want to understand this part, first of all, you need to understand this structure. Even in class 11th, morphology we discuss. This is what you need to discuss. Clear? This is what you need to discuss. So just look at this diagram. So see, this embryo is growing. It is forming different stages. It will form the cotyledons. It will be having plumule. It will be having radicle. It is having the suspensor part. So that's what we need to discuss actually. Okay, that's what we need to discuss actually. So see, ultimately growth is taking place. Ultimately number of cells are increasing. So when that number of cells are increasing, of course they are going to form some structure. You can compare it with the humans as well. In the case of humans, you know that initially we all are developed from a single cell, that is zygote. We all are developed from a single cell, that is zygote. That zygote is dividing, forming multicellular structure. You will see blastula, right? You will see morula, you will see blastula, you will see gastrula. And further organs will form, yes or no? And further organs will form, yes or no? Yes or no? But later on what is happening? Right? That layers are forming the nervous system, the digestive system, like different systems will also form. Same is the case here. Initially that zygote, the single cell was there, it is dividing. It will become multicellular and it is going to form a complete structure. Right? It is going to form a complete structure. Right, bache? So this is what you need to understand, that embryonal axis, the main axis in between, right? In between the radicle and plumule, or you can simply say plumule and radicle, is known as your embryonal axis. So this is the complete embryonal axis. That is how that embryo is growing. Clear, bache? That is how your embryo is growing. Bache, ma'am, aapko rakhi kisne bandi? Soumyadev, I don't think that this question will come in the NEET examination. Hai na? Done. So see, when you look at these dicots, see, why do we call them as dicots? First of all, look at this part. These are the two cotyledons. These are the two cotyledons. Cotyledons are nothing, they are the reserved food material, right? They are nothing, they are the reserved food material. So with that growth, right, the zygote is going to take the nourishment from the endosperm. So during that developmental stages, right, during that developmental stages, that zygote will take all the nourishment from the endosperm. Right, that will utilize all the endosperm. During its growth period in dicots, the embryo will take all the endosperm. It will take nourishment from all the endosperm and it will store it in the form of cotyledons. Right, it is going to store it in the form of cotyledons. Are you getting my point? Are you getting my point? So there will be how many cotyledons? 2 cotyledons will be there. Right. So now just imagine this part. Okay. Just imagine this part, that when in the case of dicot embryo, I'm saying that this dicot embryo during its growth, it will utilize the complete endosperm. Right. It will take all the nourishment from endosperm. It will utilize the complete endosperm. So obviously it will also keep some reserve food with it. Like let's say I gave you a chance that yes, I have certain amount of money. You use it. You can take it, and you are like, I'm going to take the full amount, hai na? So obviously you are going to save it. It's not like that you will just take that money from me and you will just use it, hai na? It is not like that. Of course, you will keep it for yourself for the future as well, isn't it? Isn't it? Same is the case here. So when that embryo was growing, okay, when that embryo was growing, so what they did, when that embryo was growing, what they did, they just used that endosperm, and in the embryonal axis itself they kept that cotyledons, okay, which is having the reserve food material. Now cotyledon, like these arms of mine, these are cotyledons. So something is above cotyledon. Can I use the word epi? Epi means above. Epicotyle, above cotyledon. Can I use this word epicotyle? Above cotyledon. Something is below cotyledon. Can I use the word hypocotyle? Hypo means below, epi means above. Right? So there is nothing to cram here. You have 2 cotyledons, reserve food. Above cotyledons means above epi, below hypo. Epicotyle, hypocotyle. And then this epicotyle here. See, here it is having what? It is having the plumule. And this radical hypocotyle will, this part, it is the radicle. So that's why we are saying the axis in between this plumule and radicle. The axis in between this plumule and radicle, it is the embryonal axis having these 2 cotyledons. Clear, bache? This is the embryonal axis. Is that clear? Is that clear? And let me mention it here that this plumule will form future shoot. I hope you remember this. This radicle will form future, R for radical, R for root. So it will form future root. Clear? It will form future root. Done? So plumule will form future shoot, radical will form future root. So now just imagine. Now this is the dicot embryo. Right? This is the dicot embryo. Now this diagram. Yes. So students, they get confused here. That ma'am, okay, you were showing it something like this. Now, what is the structure? They confuse the naming as well. So it's very simple. See, these are the 2 cotyledons. This part above the cotyledon is epicotyle, and this portion is going to be plumule. Yes or no? Yes or no? This part, these are the epicotyles. This part is known as what? This part is what? It is the epicotyle. This is cotyledon. These 2 arms are cotyledon, right, attached to the embryonal axis. This part is the epicotyle, and this terminal part here is the plumule. Am I right? This lower portion is the hypocotyle, and then the feet, you can consider them as the radical. Clear, bache? So if I will keep my cotyledon like this, if I am going to bend it like this, right? So I used to call it as yoga class basically. See, yoga part, it is basically yoga part. So see, cotyledon, so you are bending it like this. So if I'm bending like this, obviously this plumule part will come down, yes or no? This epicotyle part will go like this. Imagine it now, epicotyle part will go like this. See, this is the part. So here actually this dicot embryo is doing that yoga. Okay, so the cotyledons which were like this, it is going to fold them in this way. This portion, that's why here is plumule. Here you will see that, right, this portion here is the plumule, and this part here is the radicle, and radicle which will form future root is going to, it will be covered with the root cap. Clear bache? It will be covered with the root cap. Done bache? So this is the structure of your dicot embryo. Clear? It is the structure of your dicot embryo. Done bache? Next to it is the monocot embryo. Now when it is the monocot, only one cotyledon is there. Only, mono means one. Only one cotyledon is there. So in this particular, here in this particular embryo, you will only see what? You will see only one cotyledon. So don't you think, like see, I have one embryo here having two cotyledons. Clear? So that is why there is no need to have the endosperm here in this particular seed. Ultimately, where this process is taking place, it is taking place in ovule. Ultimately, this embryo sac is present in ovule. So after fertilization, your ovule will form seed. Your ovary will form fruit. So if that process is taking place in the ovule, which is the future seed, so ultimately it is the seed which contains embryo, yes or no? Ultimately, it is the seed that contains embryo, yes or no? Yes or no? Ultimately, it is the seed that contains embryo. So when it is a dicot seed, when it is a seed of a dicot, clear? So of course it is having two cotyledons. So in that seed, no endosperm will be there. No endosperm will be there. Why? Why? Because ultimately endosperm is what? It is the nutritive. It is the reserve cells having reserve food. It is the tissue having reserve food, right? But that reserve food is already stored in the form of cotyledons. It is already stored in the form of cotyledons. So can I not say that when we talk about the dicots in their seed, you will not see endosperm? No endosperm will be there. So mostly in the case of dicots, when you talk about their seeds, you used to call it as X-albuminous seeds or non-endospermic seeds. Right, X-albuminous seeds or non-endospermic seeds. But it's making sense, yes or no? Already one embryo is having the reserve food material. So what is the requirement of keeping that endosperm there? There is no need for that. It has already loaded its reserve food in the cotyledon. So there is no need for that. Right? So that is how you can remember that when you talk about the dicots, right, their seed is also known as X-albuminous or non-endospermic, right? In majority of dicots, that's what, that type of seeds are present. Exceptions are there, right? If I talk about the exception is your castor. Castor being a dicot, still it is having endospermic seeds. Castor being a dicot, still it is having endospermic seeds. Okay, castor being a dicot, still having endospermic seeds. Clear? Clear? This is the exception. And now when you talk about the monocots, mono means single cotyledon. So if let's say it is a monocot seed, if it is having only one cotyledon, don't you think it is unfair for this embryo? It needs more nourishment. So don't you think that here the seeds can be endospermic? The seeds will be endospermic or albuminous. Or albuminous, yes or no? Seeds will be endospermic or albuminous. Of course they are. है ना? Of course they are. Without any doubt they are. Yes or no? है ना? So there is no need to cram it. It's simple. If two cotyledons are already there in embryo, there is no use to keep extra reserve food material in the form of endosperm. Seeds are going to be non-endospermic. If there is only one cotyledon there, of course it needs more nourishment. The seeds are going to be endospermic, albuminous there, right? So here when it comes to the exception of dicots, your castor being a dicot, still having endospermic seed. And in the case of monocots, it is the orchid Orchid being a monocot, still having non-endospermic seeds. So these are the exceptions here, right? These are the exceptions here. So now just look at this diagram, just look at this part. So here, bache, can you see the monocot embryo? Can you see the shield shaped structure? This is your scutellum, right? Here this cotyledon is the shield shaped. Okay, it is present on the lateral sides. See, it is present on the lateral side. It is the scutellum. What is the word? Scutellum. This is important. And here this epiblast is nothing, it is the reduced cotyledon. Only one cotyledon is there now, so it is the reduced cotyledon. Done? So this is the example of the monocot embryo. Clear, bache? So shield shape present on lateral side. This cotyledon is scutellum. Epiblast is the reduced cotyledon. Rest plumule, which will form the shoot tip, shoot apex. Radicle, which will form the root tip. It is covered with root cap. But here two extra sheaths are there. One is your colutile and another is your colurhiza, right? So see, P for plumule, colupetile. This is how you can remember. P for plumule, right? Just to remember this part. Colupetile, P is there. So plumule is covered with that P. So that plumule is covered with a sheath-like structure, colutile. Radicle is covered with colurhiza, RR. RR, that's how you can focus. Clear? RR, right? Right. So this is about the embryos here, and even bache, I have given you the idea of the seeds as well. Right? That what type of seeds are present in monocots and the dicots. And let me tell you that sometimes even nucellus is persistent. Sometimes even in seeds, nucellus is persistent. Nucellus is not destroyed. Sometimes in seeds, you will even see the nucellus. Such seeds are known as perispermic seeds. Right? Such seeds are known as perispermic seeds. You have the example of your beetroot. You have the example of your black pepper. Clear? Where seeds are perispermic. What is the meaning of being perispermic? That they are having the nucellus as well. Right? They are having the nucellus as well. Is that clear? Is that clear? Tell me. So in grass family, in monocots, cotyledon is shield shape, present on the lateral side. It is known as scutellum. Okay. Okay. So undifferentiated sheaths are also covering these plumule and radicle. Done, bache? Clear? Clear, clear, clear? Done? So endosperm may be either completely consumed by developing embryo. So dicots में examples are pea, groundnut, beans. Before seed maturation, endosperm is completely utilized, or it may persist in mature seed, like your castor and coconut. Right? So castor and coconut is the example given here. Clear? Examples are important. Right? So split open some seeds of castor, peas, beans, groundnuts, right? Fruit of coconuts. You will find the endosperm. Clear, bache? You will find the endosperm. So now they have given you one example that find out whether the endosperm is persistent in cereals like wheat, rice and maize. So that's your homework. That what type of seeds are there in wheat, rice and maize. That's your homework. Clear? So here you guys can see the embryo. Embryo develops at the micropylar end. We know it, right? Where zygote is situated. So zygote will divide only after the certain type of endosperm is formed. So this is an adaptation which will make sure that embryo will get the nourishment. Clear, bache? Clear? So this is embryogeny, the formation of embryo from the zygote, isn't it? Isn't it? So initial stages in both monocot and dicot, they are same. So zygote will give rise to, see I told you na, PGHM. So your zygote will form proembryo, then it will form globular, heart-shaped and mature. So you should know the sequence here. Fine, you should know the sequence here. It can be asked as a question in the paper. Done, bache? So typical dicot embryo, you know that it is having embryonal axis. For embryonal axis, we also use the word tigellum. This is extra, but you can note down. Okay, and it is having two cotyledons. So what will you see in embryo? You will see the embryonal axis and the cotyledon. So the part of embryonal axis above the level of cotyledon is epicotyle, which terminates with the plumule. The cylindrical portion below the cotyledon is hypocotyle, that terminates with the radicle. So radicle is also covered with the tip, that is your root cap. Its tip is also covered with the cap, that is root cap. Is that clear? That's what bache we have discussed. So in monocots, you know that in grass family you have scutellum shield shaped cotyledon present on the lateral side. Clear bache? Present on the lateral side. Done bache? Done. Sure. So next part is the seed part here. Okay, next part is the seed part here. We can finish it today before my class. So let's speed up bache. So seed, you know that it is the final product of the sexual reproduction. See, let me mention few changes here, right? Post-fertilization. See, let me mention some post-fertilization changes here. Okay, so ovule will form seed. Ovule will form seed. Am I right? Ovule will form seed. Yes or no? Yes or no? So integuments of ovule, the integuments of ovule, they will form the covering of the seed, that is seed coat, right? So here you can mention outer integument will form testa, which is the seed coat. Inner integument will form tegumen, which is the inner seed coat. Is that clear? Yes, bache. Is that clear? Yes. So the ovary will form fruit. Ovary wall will form fruit wall, that is your pericarp. These are the post-fertilization changes. Yes, these are the post-fertilization changes. Yes or no? Yes or no? So rest, like you know that the synergids will degenerate, antipodal cells will degenerate, right? The hilum, do you remember that funicle? That funicle of ovule, that will also form that stalk which will attach seed to the ovary, right? Which will attach, seed to the fruit, right? It will be like that. That hilum, that scar, that will still be there on the seed. That micropylar end will remain like that. So, which will be used for the exchange of gases. So, these are the post-fertilization changes here. Clear, bache? These are the post-fertilization changes here. Is that clear? Is that clear? Yes or no? Fine. Done. So that's what you need to understand. That's what you need to remember. That's what you need to revise again and again and again. Okay. Okay. So next to it is the seed. I already told you about the seed, but let's revise it very quickly. So that's again your homework. You will write a note on endospermic and non-endospermic seeds. And examples are very important here. Clear, bache? Examples are very important here. Done, bache? So fertilized ovule is what? It is the seed. So seeds are formed inside fruits in the case of your angiosperm. So you know that typically a seed consists of seed coat, that is covering the cotyledons and the embryonal axis. This is what we have in a seed. The seed coat, cotyledon and embryonal axis. So cotyledon, you know that simple structures which are swollen because they have reserved food material, right? Like in legumes. So mature seeds, they may be X-albuminous or non-albuminous, or right, that they do not have the endosperm, like in the case of your dicots, pea groundnut. So albuminous seeds are the one which are having the endosperm: wheat, maize, barley and castor. The examples are given. And sometimes, as I said, like black pepper and beet, they are even having the nucellus persistent, so that is perispermic seeds. So we have already covered this part. Clear, bache? Clear? And I told you that integuments will form the seed coat. Micropyle will remain as a small pore in the seed coat, which will help the exchange of oxygen and water. Done, bache? So as seed mature, its water content is reduced and seeds become relatively dry. And you know that seeds are very important for the dispersal. So as seed mature, its water content will be reduced and seeds will become relatively dry. Clear? So general metabolic activity will also slow down. So this stage is what? It is the state of inactivity, dormancy. Right? So when seed is not getting the favorable conditions, it is in dormant state where metabolism is less, inactive it is. Clear, bache? Right? So when conditions will be favorable, it will germinate. Fine, it will germinate. So you know na, ABA plant hormone, abscisic acid plant hormone, the one which will maintain that seed dormancy. Your gibberellin will break the seed dormancy. Remember bache? Yes. So you know that now ovules will mature into seeds, ovary is going to form the fruit. So transformation of ovule into seed and ovary into fruit will be there, right? So the wall of ovary, I told you, it will form the fruit wall, and we will call it as pericarp. Remember, we will call it as pericarp. Remember, so you know that fruits can be fleshy, like in guava, orange, mango. Remember mango is a drupe. Remember, mango is a drupe. Class 11th morphology, mango is a drupe. So here pericarp, in the case of mango, it is of three types. It is divided into three parts: epicarp, mesocarp, and endocarp. Do you remember this or not? I'm talking about the mango here. In the case of mango, which is a drupe, pericarp is divided into three parts: epicarp, mesocarp, endocarp. And it is the mesocarp. It is the mesocarp which is the part that we eat. Endocarp is the stony part. And the outer covering of that mango is epicarp. Remember? Right? It's a class 11th topic. Done, bache? And sometimes your fruits are also dry, like your groundnuts, mustard, right? Some. Clear, bache? So many fruits have evolved mechanism for the dispersal of seeds, right? So this is all about the seed and the fruit. Fine? Fine. So bache, here what you need to remember? You need to remember two things. Just some fruits are true fruits, right? True fruits means it is developed from the ovary. When ovary is making the fruit, it is going to be the true fruit. But sometimes so you can also call it as eucarpic, right? So you have false fruit also. What do you have? You have false fruit also. You will call it as pseudocarpic. What will you call it? You will call it as pseudocarpic. So here you have a trick. Ambika Sharma cute. Okay, that's the trick. Ambika Sharma cute. So A stands for apple here. Fine. S stands for strawberry here. And C stands for cashew here. Clear? So these are the examples of the pseudocarpic fruits. False fruit. Here it is not the ovary that is forming the fruit. Just your apple. Apple is what? Apple is the thalamus. It is the thalamus of that flower which is forming the fruit. Is that clear? It is the thalamus of the flower which is forming the fruit. Is that clear? Yes, bache? Is that clear? Right? So this is about the fruit. This is about the types of the fruit. Clear, bache? This is about the types of the fruit. Done? Done. True fruit, false fruit. So right now I'm telling you about the false fruit. Okay, false fruit. Done? And you know that we have parthenocarpic fruit as well. Do you know that? Parthenocarpic fruit. See, bache, fruit is formed without fertilization. So here what is the scenario? Fruit is formed without fertilization, right? Fruit is formed without fertilization. So basically, of course, like let's say externally you are providing the hormones. Fertilization is not there. No fertilization means no seeds. That fruits are going to be the seedless fruits. What type of fruits they will be? They will be the seedless fruits, right? Such fruits are parthenocarpic where fertilization is not there. Okay, like you know that you have seedless banana is there, seedless watermelon is there. There are many examples. Seedless grapes are also there. Clear, bache? Clear, bache? Okay, so that's what you need to remember here. The false fruit, the true fruits, and the parthenocarpic fruits. And look at these diagrams also. Fine. Look at these diagrams also. So see, this is actually the castor. So castor is a dicot, but dicot is having the endosperm. That's an exception that you need to remember. And I already told you about the orchid, right, which is a monocot, but still having non-endospermic seeds. Done, bache? Still having non-endospermic seeds. And here you guys can see this maize. So this layer is your alluron layer, right? It is proteinaceous. It is proteinaceous. Clear? So this layer, can you see this? This is your embryo, right? Plumule, colutile, radical colyoriza. Here you will see the scutellum. So endosperm and this embryo is separated here with a layer. This layer is alluron, and alluron is also triploid in nature. Do you know that? Right? So endosperm and embryo, they are separated by this layer which is known as alluron. It is also triploid in nature. Clear? It is also triploid in nature. And I told you about the parthenocarpic fruits as well, right? So banana is such example. Done, bache? Done, bache? Banana is such example. Done? So you know that seed is very important, right? Seeds are important for the dispersal. No seeds means how will you propagate that particular plant then, right? So dehydration and dormancy of mature seeds are crucial for the storage. It's not like that always the seed will be in the active state. No, seed means it is not getting the favorable condition. Once it will get favorable condition, it will germinate. So hormonal part is there, right? And moreover, see, the dehydration and the dormancy, it is very important, right? It is very important for the storage of the seeds. Clear, bache? It is very important for the storage of the seeds. So this period can vary. Like, see, how long do the seeds remain alive when after they are dispersed? See, this period again varies greatly. Like in some few species, I told you before, na, seeds loses viability within a few months. Like we talked about the pollen grain viability. Here we are talking about the seeds viability, right? Right. Here we are talking about the seeds viability, right? So seeds of large number of species, they can live for several years, for hundred of years, for thousands of years. So here you have one example, that oldest is that of lupine, that is your Lupinus arcticus, right? This is important, bache. This is important MCQ. It is excavated from the Arctic tundra and the seed germinated and the flowered after a record of 10,000 years. Imagine. So it can remain dormant for 10,000 years, right? Your Lupinus arcticus. Okay, your Lupinus arcticus. And the recent record of 2,000 years of old viable seed is of date palm, that is your Phoenix dactylaria. Done. So these examples can be asked, right? So it is discovered during the archaeological excavation at the King's Harrod Palace near the Dead Sea. So this can be asked. So you know that pollen grain viability also vary, seed viability also vary. So seeds can remain dormant for many years as well. Okay, so they can remain dormant for many years. Okay, done, bache? Done. So do read it from NCERT, do revise it. So this is the last topic. Let's finish it very quickly and then we will move to the next part, that is apomixis and the polyembryony. See, if I have to define apomixis, I will tell you two lines. No meiosis, no fertilization. No meiosis, no fertilization. That's how I'm going to define apomixis. Actually, apomixis is a form of asexual reproduction. It is not actually the sexual reproduction. It is a form of asexual reproduction. Why? Why am I saying asexual reproduction? Why am I saying the asexual reproduction here? Because there is no meiosis, no fertilization. So it is a form of asexual reproduction that mimics sexual reproduction. That is going to copy sexual reproduction, but it is actually not the sexual reproduction because there is no meiosis, no fertilization. Apomixis, bache, it is used for making the seeds, apomictic seeds, right? See, imagine this part. Try to understand this part. Seeds are the product of fertilization. When we are saying parthenocarpic fruit, we are saying the without fertilization fruit is forming. Still, you can apply hormones externally, you can do this. Here you are making seeds. Seeds are actually the last product, the final product of fertilization, but still, here we are forming the seeds without meiosis, without fertilization. So it is actually a type of asexual reproduction that mimics a sexual reproduction, right? That mimics a sexual reproduction. Here everything is going to be same. Like here you will see the formation of the embryo sac. You will see a zygote, diploid zygote will form. You will see that diploid agapparatus synergids will be there. You will see that diploid antipodal cells will be there. And here also you will see diploid, two diploid polar nucleus. Two diploid polar nucleus you are going to see, right? This is what you are going to see here. So it is actually totally, it is copying what? It is a mechanism, right? It is a mechanism, apomixis. So apo means without mixing. Mixes means mixing, right? So there is no fertilization, there is no meiosis here, right? So it is the form of asexual reproduction that mimics the sexual reproduction. Fine, bache? Fine, bache. So it is very important for the quick multiplication and production of the genetically similar plant from a single parent. Okay. So apomictic seeds are formed. Apomictic seeds are formed where it will be actually like sexual reproduction, but it is not. So see, some species of Asteraceae and grasses, they have evolved a special mechanism to produce seeds without fertilization. So it's a special mechanism, that is apomixis. So apomixis is a form of sexual reproduction that mimics sexual reproduction. It's a form of asexual reproduction that mimics a sexual reproduction. Right, bache? There are different ways of making apomictic seeds. In some species, diploid egg cell is formed without reduction division and develops an embryo without fertilization. Right? Right, so apomixis, it is very important. Just let's say you have created a genetically advanced plant. Genetically, let's say you have created a transgenic plants, right? Transgenic plant. Okay, so you have added the different characters there. So from that transgenic plant, you have created it once. Every time you are not going to perform that techniques. Every time you are not going to use that techniques. Every time it will not be the techniques of biotech that you will be using, the techniques of recombinant DNA technology or whatever it is. You have created one, you know, genetically advanced plant. You have created that hybrid plant. Now what can you do? What can you do? Think about it. You can use, right, you can make the seeds of that particular plant. You can use this phenomena apomixis. You can go for this type of reproduction. So for the farmers it is very useful. They will, right, so they will create the seeds and they will grow the new crop, isn't it? So it's useful as well. So it's an asexual reproduction that mimics sexual reproduction. And when it is polyembryony, many embryos. So usually in one ovule you will see one embryo. In one embryo sac you will see one embryo. But sometimes what happens? The surrounding cells, let's say the cells of the new salus, they are also growing within the embryo sac. They have formed many embryos. So in citrus and in mango varieties of some of the new cellular cells which are in surrounding of that embryo sac, they start dividing. They will protrude into the embryo sac. They will enter there in the embryo sac. They will develop the embryo. Right? So in such cases, each ovule will contain many embryo. That's polyembryony. Clear? Polyembryony. Right? So you will tell me the examples, more examples of polyembryony. You will tell me more about the apomixis. So that's it. Right, bache? So here you can see hybrid varieties of several of our food are being cultivated. Right? So cultivation of hybrids has tremendously increased the productivity of cost. Right? So one of the problems of hybrid is that hybrid seeds have to be produced every year. So if seeds collected from hybrids, they are sown, the plants and the progeny will segregate. They do not maintain hybrid characters. So in that case, you know what we can do? Apomixis is a blessing. There is no segregation of characters. Like you have created a genetically advanced plant. You do not want, right? You do not want it to get mixed with any other character. Right? So in that case, you need seeds, but that seeds should not, because if the seed formation is there, means meiosis will be there, fertilization will be there. So obviously characters, the variations will be there. We don't want that. We want to propagate that particular plant as such. So in that case, apomixis is going to help. Okay, so this is all about this chapter, bache. Finally we have completed this chapter. Soon I will keep the session where we will practice top 50 questions of this particular chapter, that is sexual reproduction and flowering plant. Now tell me which chapter you want to start next. Right, it will hardly, I always used to say it is going to take here 10 seconds. Do let me know in the chat section, bache, in the comment section, that you liked the session or not, and do let me know that which chapter you want to start the next. Right, so from coming Monday accordingly, right, I'll schedule that particular chapter then. Clear, bache? Clear, bache? So do revise it. Okay, do revise it. Make the notes. I'll share the notes in our official Telegram group, and here you guys can check the videos. All are useful for you. Fine, fine, and do check this particular video, the roadmap. The roadmap to score 650 marks in 239 days, bache. Right, it is an eye-opening video. You will realize the time that you have and how to utilize that time. Okay, okay. So in the comment section, do let me know, bache, you like the session or not. And as I said, tomorrow at 6, yes, at 6 p.m. See, breathing and exchange of gases, one shot will be there. So do attend that class. Okay, do attend that class. So thank you so much for watching.